All right, we're going to go ahead and get started. Hello, everyone. Whoop. So before we do get started, I want to let our audience know that we are recording today's webinar. It'll be available for viewing in a few days, and you'll be able to find it on the NOAA One Health and NOAA Regional Collaboration websites. So thank you all so much for joining us today. I'm Catherine Egan. I'm the acting NOAA Southeast and Caribbean Regional Coordinator. Today's webinar is coordinated through the NOAA Regional Collaboration Network and NOAA One Health. The NOAA Regional Collaboration Network is a network of employees across NOAA, connecting our work to improve NOAA's services to the nation. The regional teams around the country periodically coordinate events like today's webinar to educate NOAA staff and partners about NOAA's programs. All right, so before we do get started with the webinar, I'd like to run through a few webinar housekeeping items. So first, if for some reason you are listening to me talking but cannot hear anything, here's the information on where to get the audio. So you can tune in via your computer's audio or via your phone, and you can access that via the audio pin located here in the audio drop-down menu. So like I said earlier, our webinar today is being recorded. So please keep your cameras off unless you are presenting and prompted to do so. So as participants, you are all joined in mute status. There'll be an opportunity for you to type questions in later. I'll touch base on that in a minute. And like I said, we please ask that you leave your webcam off as the webinar is being recorded. So like I said, I'll be monitoring questions in the questions pane during the webinar. And so I really encourage you all to please ask questions throughout the webinar, and then we'll do a Q&A session with our presenters at the end of their presentation. All right, so with that, I'm now going to turn it over to Morgan Zabo to tell you about NOAA One Health.
Thanks so much, Catherine. Hey, everyone. It's a pleasure to be with you all today. My name is Morgan Zabel, and I work in NOAA's Climate Program Office and help to coordinate the NOAA One Health group. So before we get started with our featured presentations today, I want to give you all a background about One Health and the upcoming NOAA One Health Summit. So if you're unfamiliar with the One Health concept, One Health applies a collaborative, multidisciplinary, and cross-sector approach to address potential or existing risks at the interface of humans, animals, plants, and ecosystems. The One Health concept recognizes that the health of humans is linked with the health of aquatic organisms, plants, and the environment. And the work within One Health creates conditions for economic vitality and growth and supports scientifically sound, economically sustainable, and just practices and policies. And NOAA has a One Health team and will be hosting the first NOAA One Health Summit in Washington, D.C. from August 15th through 16th. The NOAA One Health Summit in August will highlight and bring together scientists and practitioners across the agency to connect and highlight NOAA's work on health and related issues and with the health sector that supports the One Health approach. Registration is now open for the summit, and you can find out more information available on the NOAA One Health page. The agenda for the summit will be available soon. And in preparation for the One Health Summit, we're hosting webinars to highlight a single aspect of the health work being done in the 8 NOAA regions. Today's webinar is the 6th in that series and is done in partnership with the Southeast and Caribbean region, where NOAA staff will discuss marine mammal health. While we're highlighting this aspect of health work in this region, please be aware that there are examples of almost every One Health thematic area in every region around the country. And we hope that you can join us for the remaining webinars in the series between now and the end of the month, and the beginning of August. And of course, we hope that you can join us for the One Health Summit, either in person in DC or virtually. And now I'll turn it back over to you.
Thank you so much, Morgan. Really appreciate the primer on the One Health One Health Summit coming up. So with that, I'm really excited to welcome our 2 speakers here today to discuss marine mammal health. Our first speaker is Wayne McFee. Wayne McFee is a research wildlife biologist at the NOAA National Ocean Service's Hollings Marine Laboratory and has been the program lead for the Coastal Marine Mammal Assessments Program since 1993. Mr. McFee conducts research on marine animal health, stranding demographics, causes of mortality, entanglements and ingestion of marine debris, age and growth, reproduction, skeletal pathology, and fisheries interaction of commonly stranded marine mammal species in South Carolina and in the southeastern US. He was also a member of the Southeast Implementation Team there we go, the Southeast Implementation Team for the Recovery of the Northern Right Whale from 1994 to 2008, and currently sits on committees that investigate unusual mortality events, marine mammal entanglements, and marine debris related issues. Our other speaker today is Bonnie Ertel. Bonnie is a marine scientist with a specialty in plastic pollution. She graduated with her master's in biology from the Citadel, is pursuing her PhD with the University of South Carolina, and is currently on contract to NOS, National Centers for Coastal Ocean Science, with CSS. Her research focuses on investigating microplastics in the gastrointestinal tracts of marine mammals. So just want to reiterate again to our audience that if you have questions throughout the webinar, to please type them into the questions pane during the discussion. So with that, Bonnie and Wayne, I'm going to pass it over to you.
All right, great. Can you see everything and hear me okay?
Yep, sounds good.
All right. Well, thank you for inviting us to speak here. We're going to try to tie in some of our marine mammal health and monitoring assessments with the One Health initiative. And these first few slides are courtesy of Lori Schwacke, who used to be our program lead here at NCOS and then moved over to the National Marine Mammal Foundation, is now with the Marine Mammal Commission. I'm going to describe a little bit of our marine mammal health research in Charleston, and it kind of started in the early 1990s when we were doing stranding investigations, life history studies, and so forth to help nymphs in their unusual mortality investigations, and also for stock assessment research when we started to do some capture release programs. So it was mainly a resource focus at that point. And then around the early 2000s, we started focusing on using marine mammals as sentinels of our coastal ocean habitat and trying to tie in the animal health with ecosystem health and human health. So basically what we're talking about is the Oceans and Human Health Initiative. That was part of the Center of Excellence for Oceans and Human Health, OHH, also organizing the Cooperative Center for Marine Animal Health, and organizing the Health and Environmental Risk Assessment that we were in partnership with NIMS and Harbor Branch Oceanographic Institute. In 2010, I think we all know about the Deepwater Horizon oil spill, and that was the first time that marine mammals are actually part of the risk assessment, injury assessment, or NERDAs, as part of that investigation. And so we have continued to do resource and sentinel focus up until today. And so we'll talk a little bit more about that right now. But there's a lot of threats to marine mammals in the southeastern United States, fishery interactions, ship strikes, human interactions. But there's also a lot of these threats that are similar to or can affect human health as well. Coastal pollution, climate change, harmful algal blooms, diseases, and some of the diseases these animals carry can be zoonotic as well. How we assess the impacts of these stressors is difficult to do since we really can't bring these animals directly into the lab. So we developed an assessment toolbox for cetaceans on how to handle these animals. And basically, we want to know where the source is, the exposure to them, and then to the effect of what that exposure is to the animals. And we do that in a number of different ways through visual documentation, tagging, which can lead to distribution and movements, and then exposing the exposure being prey, environmental sample analysis, tissue analysis, which can either come from remote biopsies or capture release programs or from stranded animals. And then the effect, analyzing these tissues from necropsies, from those capture release health assessments, all into developing statistical modeling for epidemiology studies to determine the population risk and or for ecosystem modeling as well. So in Charleston, we have a resident population of dolphins here that have been studied for the last 30 years. We use an integrated study approach using the stranding investigations, longitudinal photo ID, remote biopsies, and capture release programs. All of this contributes as an additional reference populations for studies that I'm going to talk about in a second, to look at young of the year, adult survival rates, pregnancy success, pathology baselines, and then also to distinguish the coastal and estuarine stock overlap and interactions. Then we also analyze the reproductive seasonality from strandings and from photo ID. And then finally, looking at the influence of land use on chemical contaminant exposure. So in the Southeast US, we've done a number of capture release studies all around the Southeast for stock assessment, also for health assessments and for unusual mortality events or national resource damage assessments. And we've also done remote biopsies in a number of places around the Southeast as well, mainly for contaminant and genetic studies. So all of this is in a strong collaboration with a number of people from the National Marine Fishery Service, state agencies, and so forth. And so this was by doing these capture release programs and biopsy studies has provided a health reference values for a number of different parameters, including body condition and blood parameters, and also helping us understand spatial trends in a broad range of contaminants and pathogen exposures, such as Brucella, which was a big problem in 2012. But it has allowed us to get baseline levels for PCBs and other contaminants, other organochlorine pesticides. As you can see in this map over here, that I guess it doesn't work, does it? Hang on one second. It gives you kind of an idea of where the hotspots are for, if you can see the red dot on there, the Brunswick area is very high in these animals and PCBs. And has allowed us to do further research in those areas as well. Don't seem to be, I can't seem to advance the slide. Maybe we get rid of this. How do you get rid of this? Let's see. Okay. Well, by doing all these captures, we've, you know, since 1995, this is actually was a big help when DWH happened. It was, you know, critical to understanding the effects of DWH on these animals. Combined with the unusual mortality of event findings that were going on at the same time, the results from our studies there showed chronic poor health, failed pregnancies, increased mortality of dolphins in the aftermath. These findings have informed planning for restoration projects to promote stock recovery, and actually the group from the National Marine Mammal Foundation and other folks have gone back down to that area and in the last couple of weeks to continue studying the effects of Deepwater Horizon and other freshwater input that is potentially going to be going on there as well. So in summary on this part, there's the integrated live animal health studies of significantly advanced science for injury assessment, but that the existing and emerging stressors along the southeast coast require consideration of cumulative effects, and they need new, innovative, prospective risk assessment approaches. These new approaches also need to expand baseline health data for coastal and pelagic tertiop stocks and other cetacean species as well. And I'm going to talk to you about a couple of new approaches that we're doing here now in Charleston, and then we'll move on to Bonnie. But one of them is using scanning electron microscopy with, if I can get this right, electron dispersing X-ray spectroscopy technology, or SEM-EDS, to detect elements that are common in crude oil that may be in the teeth of animals that may be indicative of oil exposure. This will hopefully give us a better feeling as to when these animals were exposed, because teeth give you a yearly, basically a yearly look at what they were exposed to. And it may also define geographical areas of concern. We're also using UAS, or drone assessments. We've had a couple of studies now looking at low salinity habitat usage, because we've had a lot of animals recently, not just in Charleston, but in other areas of the southeast, that are seemingly moving upriver into lower salinity water. And so also looking at body condition for those animals that are in that area. And also another study looking at blow microbiome sampling, which I'll get to in a second. So I'm going to start with the SEM-EDS tooth study that we are doing. We basically are looking at 3 different areas. One is the northern Gulf of Mexico, with that big oval on the left-hand side of the screen, that we are collecting teeth from animals that have stranded there prior to DWH, during DWH, and after DWH. And also looking at the same thing for animals that are stranded in Sarasota that were, we think, not affected by the oil spill, but are in the same basin as the animals in the northern Gulf of Mexico that were exposed. And then looking at another reference site in Charleston, or in South Carolina, where we think animals aren't exposed to oil exposure. And so essentially what we are doing is taking these teeth, cutting them in half. This is the SEM-EDS instrument on the left-hand side, and then just a picture on the right-hand side of the tooth laying on one of the stages in the SEM-EDS. Once it's in there and it's closed up, we've got electron beams that are placed over the tooth, run across the tooth, and it scatters what different elements are picked up in the tooth. And basically what you end up with is a picture of different elements that are color-coded on the screen. These happen to be vanadium and nickel, which are in high concentration in crude oil. And so, can anybody tell me, can you see my pointer here?
Yep, we can see it, Wayne.
Okay, so this black area here is the pulp cavity of the tooth, and there's no green dots in there, which is suspected, but the area outside of it, there's a lot of green dots going on in there. That's nickel and vanadium that has showed up, which could be an indication that this animal was exposed to oil at some point. It's interesting for us to know whether or not these animals, or when these animals are actually exposed. So if this animal had just died, we would expect that we would see signatures of nickel and vanadium if it was exposed near the pulp cavity, because this is the last layer of growth that the animal will lay down before they've died. So that's what we're seeing here. So we think this animal may have been exposed to oil just prior to death. So the timing of this would be very interesting to researchers in the field. And then that is ongoing study. I'm going to move quickly on to drones so that Bonnie can get in here, but we're also working closely with the College of Charleston and with Ocean Alliance on using drones in some marine mammal research. And we've done a couple of studies with Nicole Principe and Colin Perkins Taylor in the Grice Marine Biology program at the College of Charleston. Our platforms use the DJI Phantom 4 or DJI Mavic Air 2 under a NIMS permit to fly about 9 meters above the dolphin for good body condition photos, and also at a 30-meter altitude away from these animals while we're traveling to find them. One good thing about drones is that we can pair them with photo ID studies at the same time to look at potential health and behavior. I'm going to show you a quick video, and if you'll concentrate not on these 3 animals that you see here, but you're going to see an animal pop up to your left in a second. Where'd it go? And if you look to the left of these animals, you'll see an animal pop up with trailing bubbles. You see them right there. Okay, so when we saw that video, that's kind of an indication to us that an animal is not in very good shape. When we looked at the photo ID picture from the boat, we noticed that this same animal taken at about the same time, it's a little difficult to see in this picture, but it has a little bit of a peanut head, a depression behind the blowhole, which is an indication to us that this animal is not in very good condition as well. So the combination of two of the drone video and this still image corroborates that that animal is in poor health. So that's just one aspect that we can use drones for. We're also using drones for microbiome analysis of the cetacean blow, and this has been done in the past for large whales that have a very large spout or mist that is made during exhalations, sometimes up to 10 feet high. They tend to linger in the air. It's easy to just set your drone on top of that blow and get a nice, good sample. For your coastal smaller dolphins that surface more erratically, the blow is not high at all, from inches to maybe 2 to 3 feet. It may be difficult to sample the blow using a drone that way. So we've tried it, and not many people have done this yet on estuarine dolphins, and we're getting some good results. But this respiratory disease is one of the leading causes of natural mortality, and when they blow, they expel mucus and moisture and microbes from the upper respiratory tract. And this blow microbiome resembles the respiratory microbiome as well, and might be able to tell us, what's going on with the health of these individuals, instead of sampling them during health assessments, which can be really stressful on the individuals, can be very expensive, large teams, a number of boats, and take a lot of considerable time and effort. Although you can, with health assessments, get a wide range of other health data to pair with lung data. And then you also have the ability to track animals over time with satellite tags. But the drones, if you want something that's quick and easy to look at an assessment of at least respiratory health, it's less stressful, it's cost effective, it's a small team, little time and effort to do that, but you do get limited samples, and it is difficult in identifying individuals from this method. But we are using Ocean Alliance's SnotBot that you may have heard of. Essentially is a Mavic drone that has Petri dishes placed on arms that are affixed to the drone. And the mist gets collected onto the plates, and then they're taken back to the lab and analyzed for the microbiome DNA. I'm going to show you a video here of how we collect these samples, and you can see the animals blowing there, and then the drone flying through the mist. The good thing about this sample that we took right here is that we are actually standing across where you see right now on land getting this. So we didn't have to take a boat out to get these samples, which is another big advantage of this as well. It doesn't like the thing. How do you? Okay. From these drones, you can also get body condition measurements, which is important to determine health as well, using morphometrics software, and also getting a body area index using Collatrix. And essentially that software will measure the dolphin at certain points and get an index that can be scored from different individuals over time or in different geographical areas to get health assessment. So those are just some ways that we are measuring health in these new ways we're measuring health in bottlenose dolphins here that can also tell us something about the environment they live. And so a lot of people took knowledge that have participated in these studies, and especially the people in the past that have worked with us all on the health assessments in the field. And so I'm going to pass this over now to Bonnie, so that she can tell you about some microplastics. Thank you.
Hi, my name is Bonnie, and I'm going to be sharing some of the research I've done on microplastics in our local population of bottlenose dolphins. I'm really excited to share with you guys at this One Health webinar because I feel like plastic pollution is a great intersection and application of the One Health concept. So we have plastic, which is made by humans and persists indefinitely in the environment, and it can definitely negatively impact both animals and humans. So I've been working on this research for the past year as a contractor with CSS to NCOS, or the National Centers for Coastal Ocean Science. And this is also part of my dissertation research with the Arnold School of Public Health. So just an overview of microplastics. You're probably familiar with them, but these are small particles ranging from 1 micrometer up to 5 millimeters, and they vary greatly in their shape, color, polymer type, and their source. So we have primary microplastics, which are intentionally created to be small and can include things like nurdles or microbeads. We also have secondary microplastics, and these come from the breakdown of larger plastic items. They can include fragments, films, fibers, or tire wear particles. And microplastics are a contaminant of emerging concern because they not only contain chemicals that are added to them to give them desirable qualities of plastic. Those include phthalates or BPA, both of which are plasticizers and known endocrine disruptors. Plastics can also contain flame retardants and a number of different chemicals. And additionally, they can also absorb contaminants from the environment, such as PCBs, pesticides, PAHs, DDT, heavy metals, and many other organic contaminants. Thus, microplastics really are a toxic cocktail contaminant, meaning they are really complicated and have a lot of different things going on with them. And since 2016, researchers with NCOS have been studying microplastics in marine mammals. So our program, led by McFee, has been doing health assessments, as you just heard, for many years, but starting in 2016, we started collecting and archiving the stomach and intestines of locally stranded animals. And this research in microplastics really started with Francesca Battaglia, who was a College of Charleston graduate student who did the first report in North America of microplastics in the gastrointestinal tract of bottlenose dolphins. So she looked at 7 animals stranded here in South Carolina. And that research then led to Tessa Pfeiffer, who was another College of Charleston graduate student, and she compared the microplastic abundance in cetaceans from different regions throughout the US. And her work found that microplastics were highly abundant in animals stranded from South Carolina, North Carolina, and Alabama, and that abundance was higher than that compared to cetaceans stranded in Maine and Washington, indicating that marine mammals here in the Southeast could be at a higher risk of being exposed to these contaminants, which leads to the research that I've been doing. So for the past year, I have been assessing the entire GI tract of locally stranded estuarine dolphins. And so we have samples, as I mentioned, dating back to 2016. I'm going to be doing this for the next couple of years, at least until 2026. So I'll have a 10-year data set with well over 60 animals. So far to date, we have 57. I'm going to share some preliminary results with you guys on the first 20 animals I've done. But I'm mainly focusing on just these local dolphins that have stranded within the estuaries here in the Charleston area, because those are part of our local stock. And to date, not many studies have looked at microplastic abundance in on a population level. So that's an overview of my project. And this is so important because studying microplastics in this local population can help us get a better idea of the stressors and health of our local bottlenose dolphins. And again, these dolphins are sentinels for environmental and mammalian health. So here is a video of me stretching out and measuring the entire length and weight of these intestines. So as I mentioned, I'm doing the entire gastrointestinal tract, and these intestines can be very long. I think the longest I've looked at from a full-grown male was about 45 meters, which is well over 100 feet. So they can be very long. I do divide it into 8 even sections to make it a little more manageable to process, but everything I'm going to share with you is the total number of microplastics in the entire GI tract, including the stomach and the intestines. And that is a little bit different from the 2 previous studies I mentioned. Francesca and Tessa only looked at the stomach and the first bit of intestines, but the entire GI tract is quite an interesting feat. And to do that, I rinse stomach and intestine contents through a stainless steel sieve and then digest any biological material using potassium hydroxide. This is a very strong base, which is going to essentially liquefy anything in the sample, which then after about a week on hot plates, that can be filtered, and that filter I will look at under a microscope. I'll first go through visually and pull things out, identifying them by their shape and their color. And then I'll do Raman spectroscopy. So Raman is a method to chemically identify the composition of a particle. So without getting too much into the chemistry with y'all, this machine, which we work with collaborators at Virginia Tech to use their Raman, this Raman machine will shoot a laser at a particle. For example, I have here a clear fiber. And that clear fiber will, most of the light wavelengths, will just be scattered away. However, some are picked up by the Raman and create these nice peaks, as you can see here. These peaks then are essentially a fingerprint for what that material is and can be matched to materials in a known database. So you can see here, the black line is what we picked up from this particle, and the red overlay is showing polyethylene terephthalate, and polyethylene terephthalate is also known as polyester. This is a type of plastic polymer that's commonly used in clothing and other textiles. So when I do the Raman, some do come back as naturally occurring materials such as cotton or chitin, and some I have to identify as anthropogenic or man-made, because you can see here this blue fiber. This is before I shot the laser, and then after I shot the laser, the fiber essentially just melted. So unfortunately, I wasn't able to get a complete match to a specific polymer, but instead it's deemed anthropogenic because if it was natural, it would not melt like that. And then lastly, I have polymer matches, and I've shown here 2 of the most common types that I've found, which have been nylon and polypropylene. So nylon and polypropylene are commonly used in fishing gear and other textiles, which is interesting that these fishing-related materials are ending up in marine apex predators. I should mention most of what I'm finding are fibers, so you can see some examples here, but I'm also finding all of the types of microplastics, including fragments and even some tire wear particles. And in terms of abundance, Here on the y-axis is the number of microplastics identified in the animal, and the x-axis are the 20 animals that I've looked at so far. And this is chronological, dating from 2016 to 2023. The biggest takeaway here is that microplastics have been highly abundant. I found an average of 1,319 microplastics per dolphin, which is much more than what Tessa and Francesca reported finding in the stomachs. But it kind of makes sense. The intestines are a very long organ and there's quite a bit of surface area there. So while it's difficult to say if these microplastics are accumulating in the animal, because this is just a screenshot, a snapshot in time of what was in them when they died, one big takeaway is that microplastics are highly abundant in these apex predators. And trying to tease out what could be driving all of this variability so far has been difficult. So on this new y-axis, I have the dolphin length, which is a pretty good proxy for the age of the animal. So these bigger, higher numbers are going to be older animals. And there has been no clear trends in looking at these different variables of age, sex, or stranding year so far. But one thing I find interesting is that some of these smaller animals, so anywhere from 90 to 100 centimeters, would have only survived a couple weeks, if not days, in the environment. However, these young animals are I'm still finding quite a bit of microplastics in them, which has really led me to wonder how they're being exposed, because dolphins don't drink seawater. So the only source of microplastics in these animals would either be placental transfer from their mother or possibly through milk transfer, again, from the mother. So that's something I want to look more into in the future. And I'm hoping that once I have a larger data set, some of these trends and variability may make more sense and complete the story. But again, this is just preliminary data, which will continue. Additionally, in the future, I want to compare how the microplastics in this inshore estuarine species or population compares to more offshore species. So we have an intern with us this summer from the University of Chapel Hill who's looking at offshore whale species. She's looking for microplastics in the gastrointestinal tracts of Cuvier's beaked whales as well as some sperm whales. I also want to see if microplastics could be translocating to other organs and what sort of subcellular effects that this exposure may have on the animals. Again, I want to look for possibility of transfer from milk or placenta. And then what I'm really interested in is looking at the quantifying the trophic transfer from fish. So these 5 prey species are the top preferred species of our local dolphins. And we've partnered with the South Carolina Department of Natural Resources to collect these fish, and I'll be looking at them for microplastics. And this kind of ties it all together and ties it back to human health, because us humans, we eat some of these same species. So again, a human-made pollutant is getting into the environment, affecting our animals, and studying these animals can be a great proxy for how things could be affecting our health as humans as well. So with that, I would like to thank all of the people who have made this work possible. NOAA for funding, Austin Gray for letting us use his ramen, and my advisor, Jeff Scott, who is the director of the NIH Center for Oceans and Human Health and Climate Change Interactions at the University of South Carolina. And with that, we can take any questions.
Awesome. Thank you so much, Wayne and Bonnie. Those are fantastic presentations. Yeah, we're happy to take questions now from the audience. We have plenty of time. So again, please feel free to type your questions into the questions pane. I'll read them out loud for Wayne and Bonnie to answer. So we did have a question that came in during your presentation, Wayne. This one is from Taylor, and they asked, and this is in reference to the contaminants part of your presentation. If applicable, how are these contaminants being actively mitigated? Are they largely entering the ocean through point sources or non-point sources?
Yeah, good question. You know, they are in some areas. In some areas, it's point sources that we know but some of it is non-point as well. But you know, the industrialized areas certainly have animals with higher contaminants, but it doesn't doesn't necessarily mean that they're not going to get contaminants in pristine areas because of movements. You know, sometimes these animals move great distances, so it kind of depends on what population that you're looking at as well. But it can be from both sources.
Great, thank you. Okay, the questions are coming in. So this one is from Nicole and it's for Bonnie. Is it possible microplastics within these organs may alter microbiome communities, ultimately affecting animal digestion and health? And then this person also says, I may have missed this. What was the cause of death of the animals you studied?
Great questions. So first of all, with the microbiome, yes, I would expect that it is altering their microbiome. We have played around with the idea of incorporating that within this study. However, it's really difficult because, to answer your second question, the animals that we're working with are stranded and dead. So we have to be very opportunistic with what samples we can get, and not all of them are always in the best condition. Some can be very decomposed. So I would love to look into the microbiome within the future, and that's a great area of research that's being developed with microplastics right now, but I don't know if I'll be able to incorporate it into this study.
Thanks, Bonnie. I got another question that came in for you. This one's from Jenny. Did I hear you say that you were finding tire fragments in the, oh, pardon me, I'm horrible with scientific names, Turrisops digestive tracts?
Yes, that is true. Tire particles is a newly developing area of research within microplastics. They are considered a type of microplastic because they largely contain synthetic materials, but tires contain a lot of other chemicals that I would not like to be within me. So finding them within dolphins is an interesting find. I haven't found that many, but I have been finding some, yes.
Thanks, Bonnie. All right, our next question is from Jeff, and they ask, oh, well, they say, thanks for the great talk, Wayne and Bonnie. And then their question is, with the different local and transient dolphin stocks that may be in an area, is it possible to identify a dolphin to a specific population based on video from a drone?
Not so much. Most of the studies for identification of dolphin is done by photo ID from a boat. And then to further complicate that, you know, you'd have to do biopsies to get skin samples for genetic work to distinguish animals from different populations, and even that is very sketchy because I don't think that's all worked out between coastal and estuarine dolphins. But what we have found from Colin's research is that he has been able to identify individual dolphins from the air based on their scar patterns on their backs and so forth. So with photo ID, you basically look at the side of a fin and get their characteristic shape to match them up over time if you keep seeing them. He's been doing the same thing from the air, looking straight down on individuals that have different scarring patterns that he can match up over time. So that seems to be promising. We didn't think we'd be able to do that, but it seems like we will be.
Thanks, Wayne. All right, I have another question that came in for Bonnie. This one's from Anya. They ask, did you quantify microplastics in the stomach apart from in the intestines in an effort to better compare to past studies, or did you just do the whole thing together?
I separated them for that exact reason, so that I can go back and compare it to what other studies have done. And I didn't include it here just because breaking it down gets a little bit confusing, but I even have them in the intestines, I separated into 8 even sections to see if there is movement throughout the intestinal tract. So I have the stomach is separate, And then the intestines gets broken down into 8 different sections. So for the simplicity of showing you guys just broad results here, I did not get into that. But that's some analysis that I will definitely look forward to doing in the future.
Thanks, Bonnie. Okay, I actually have a few questions of my own. So kind of going off Jeff's question here about the drones, this one is for Wayne. How can the use of drones and marine mammals be used for managers in NOAA's managed areas?
Yeah, what we're finding with using the drones right now with the microbiome research and flying through the blow of dolphins, we'd like to expand that to managed areas such as NEARS sites. Those are the estuarine reserves that NOAA manages. And to do sampling of dolphins in those zones as well. And because those areas are managed, we would think that they'd be a little bit more pristine and not so heavily inundated with industry, but not always the case. But anyway, compare those animals with animals from the Charleston area that we've just done. Ideally, it would be nice to hit every one of the near sites doing this and kind of rotating it from year to year to see what the change in the microbiome may be, because as temperatures climb and the climate changes, the microbiome in the water and in more than likely in air breathing animals is going to change as well. So it'd be nice to do that over time.
Thank you. That's really helpful. There's not a whole lot I know about drones, but it sounds like it can have really great management purposes. Another question came in for you, Wayne. This one's from Gina. How does the surface behavior, i.e. multiple dolphins surfacing at the same time, influence snot sample collection?
Yeah, good question. You know, we were hoping when we first started this that we could do this like a large whale and be able to do, you know, photo ID and know who the animal is so that we could easily collect samples. But it's, you know, right now we're kind of in that phase of, you know, can this actually work? And so we're not really caring too much who we get. We just want to get something. And so, it would be advantageous to know who you are sampling prior and be able to get a sample from them. For instance, there's a number of animals that that we have captured in the Charleston area during our capture release program in the past, and we've taken swabs from their blowhole for microbiome studies, and those same animals are still around out here, and we think we actually sampled one of them that we might be able to compare those microbiome samples from years ago to today. So yes, we would like to do that, be able to sample them singly, but it's actually easier to do it in a group because the problem obviously is that you don't know who you're sampling in that, but you do get a general idea of the overall microbiome of that group, if that makes sense.
Thanks so much, Wayne. All right, I have a few more questions myself to ask you both. So this one's actually about the SEMEDS, quite the acronym. Can it be used to detect barium for indication of freshwater exposure?
Yes, absolutely. It can detect you know, a lot of different elements. So barium is definitely one of them to be able to check for freshwater exposure. We're hoping to do that as well because we had some questions from our Southeast fishery science folks who were interested in that technology as well to be able to detect freshwater exposure in these animals. So yes, we will do that as well.
All right, thank you. And I do have a couple of microplastic questions for you, Bonnie. I have one that I actually hear quite a lot, and that is, should we worry about microplastics in humans?
I get asked that often, and I don't have a definitive answer right now because the verdict is still out. It's really difficult, as you can imagine, to study these things in humans because I can't get a hold of human intestines to see how many are in there. There has been studies finding microplastics in human blood, placenta, milk, and feces. So we know they're there. However, we're still trying to determine the effect. At a subcellular level, they can cause inflammation, oxidative stress, and a number of other issues within a tissue or organ system. However, a lot of these things are done in extremely controlled laboratory environments, so translating that to the real world is difficult. Additionally, as I mentioned, microplastics is such a diverse suite of contaminants. I've heard it's been said that a microplastic is not a microplastic is not a microplastic. They are all so different and can have such different effects. So it's a very complicated question to answer. I think I kind of leave it at I'd rather not have them in me. I'd rather not be asking that question. But that is something that is continuously developing within the world of microplastics. And I think within the next couple of years, we'll really have a better handle on what the effects, especially the long-term effects that these particles could have on our human bodies.
Thank you so much for that. I do have one last question to ask you that's very similar. Beyond documenting the presence of microplastics in the GI tracts of these marine mammals, do you have plans to investigate the effects that these contaminants may have on the animals?
Yeah, so that's something that we are currently in the works of figuring out, a study to look at the effects on the gastrointestinal lining. So we want to do some transcriptomics and that's basically just seeing what is being transcribed by the cells, and that can indicate things such as inflammation. So if a cell is transcribing a certain compound, that is likely indicating that these microplastics are causing inflammation within the area. So that's a field of research that we're getting into, partnering with researchers down at the University of Florida. So that's just in the works, still working on figuring out if our samples will even work with that, because we, again, are dealing with dead animals that are at various stages of decomposition. So that's something that is going to be in the works over the next couple months, is seeing if we can try and determine some of those effects within the dolphin intestines.
Great, thank you so much. I think that is all the questions I have, and it seems like that's all the questions that our audience has as well. So I'm gonna go ahead and wrap up our webinar for today. I'm gonna steal screen sharing away from you. Just give me 1 minute, because I do want to show the audience our schedule coming up for our next few webinars. So before I do that though, Wayne, Bonnie, I want to thank you both again for presenting on your work today. We really appreciate you being here and taking the time. And I also want to thank our audience members for joining in. So please be on the lookout for announcements about future webinars in the NOAA One Health series. The next set of webinars is going to be focused on the Pacific Islands, and those webinar times and dates are listed here, July 19th and July 25th. So we hope to see you at that time. So thanks everyone again for joining.